Hello everyone, welcome from my side in our webinar today. As Sarah already introduced, we will be four of us speaking with you today, and I will shortly introduce the agenda. We will talk about the importance of flow cytometry to measure cellular immunity in COVID-19 research, and then my colleagues will talk about how flow cytometry can support us. When we are looking into the involvement of flow cytometry, as you all know that during this COVID-19 pandemic, monitoring the immune response both in regards to how our immune system is affected upon infection, as well as how to modulate our immune system to fight back, has been a constant effort. So this complex cellular immune component from both innate and adaptive immune compartment play a crucial role here. Numerous studies have shown how T and B cell responds towards SARS-CoV-2 infection, and therefore is critical to study them upon vaccination to evaluate long-term memory. Here are some of the recent articles you can see as a snapshot that were reported where such analysis was used. My colleagues later will elaborate further on this topic during the talk. And as you can see, of course, flow cytometry has been played an important role for such kind of cellular analysis. So why flow cytometry is a useful tool here? I hope all of you maybe already are using this tool, and as you know, the advantages are that you can get a single cell resolution using this technology. You get to see the whole subpopulation component. It's very sensitive tool. It allows also detecting very rare cells, which is very important in the case of these kind of viral infection. It has been shown that if you are start to work with 10 ml whole blood, at the end you will be able to only detect around about 4,500 SARS-CoV-2 reactive T cells, and it's also similar in case of B cell. So when the cellular components are so rare in the sample, it's very important that you have a sensitive tool like flow cytometry. Of course, the flow also allows you to transfer your assay into a high throughput setup, and you can also perform multiple parameter by multiplexing and so on. However, this tool also comes with a lot of problems such as data variability. And the variability that arises during flow analysis has been rooted towards different factors. Of course, the first one where the variability arises is coming from the donor that you are using in your experiments, and it's obviously very commonly observed. However, very importantly, we must also remember that there are a lot of flow cytometry data variability arises from the component of the assays, such as the reagent that you use, because they are different in terms of their lot and specificity. The instrumentation also affects the data variability through different operations. And analysis, of course, involved a human factor based on who is analyzing the data. And if you don't use the right protocol, that also affects. I just quickly want to highlight here that here at Miltenyi Biotec, we take great care in terms of ensuring the reproducibility to avoid this kind of data variability in flow cytometry by our solutions in terms of reagent, instrument, and analysis tool. Of course, donor variability is something that is very difficult to address. I will just take the next 2-3 minutes to shortly introduce how we take care of the reproducibility issue in flow assays. For this, to start with, our reagents are based on recombinant antibodies that we call reaffinity antibodies, and these products offer high lot-to-lot consistency. Our antibodies gives no unspecific binding in your assay. It also allows you to save time by, as our antibodies are already pre-titrated, and you only need single isotype control. And we ensure the validation of all our flow antibodies in respect to their application. Now, looking into the instrumentation, there we also worked very hard to ensure reproducibility through our superior automation support. So our flow cytometer MaxQuant family ensure the automation for simple uses, thereby reducing operator variability. And this does not hamper the data quality, since we only allow less than 0.1% carryover among sample to sample, and we also give superior data such as absolute cell counting. We also offer consistency among instrument to instrument through multi-instrument alignment. Finally, the analysis software comes along with our advanced analysis tool that we call as Express Mode, which is integrated within the MaxQuantify software in our all-flow cytometer. This tool also ensures reproducibility through automated data acquisition and analysis, and thereby it reduces the operator-induced variability, and this can be easily integrated into your SOPs as a quick and robust analysis. So this is a very short summary how we, from Miltenyi Biotec, with our effort, we try to reduce the data variability and ensure quality data and reproducibility. Now, in the next presentation from my colleagues, you will hear more in how our tools also can help you to upgrade your analysis and your research for specifically in the SARS-CoV research context.
So welcome everybody also from my side. I will now continue with flow cytometry analysis of SARS-CoV-2 reactive T cells. So let's just continue with a brief introduction into T cells in general. So why are T cells especially so fundamental in mounting an antiviral immune responses? And so apart from the innate immune system and the B-cell mediated humoral immune responses, T cells actually play a very important or central role in cellular immune responses. So of course, all these parts of the immune system are strongly interdependent and also need to work together efficiently to generate antiviral immune responses. But why are virus-specific T cells so fundamental for an efficient antiviral immune response? Well, they not only directly kill virus-infected cells, but also support B-cell responses and thereby directly support the humoral immune response. However, most importantly, they provide long-term immunity, and this may also indicate a favorable disease progression and maybe also a better prognosis. And this is also why only assessing antibody titers either during or also after infection will not give you the full picture. And this is why we need to analyze virus-specific T cells as well. But why do we need to study virus-specific T cells, especially in COVID-19, in more detail? First of all, of course, because we just need an accurate picture of SARS-CoV-2 specific immune responses. But also research has shown that up to 30% of patients actually do not produce detectable levels of SARS-CoV-2 specific antibodies. And that especially in asymptomatic and mild cases, the antibody titers are really low and also rapidly decline. And this is actually not the case with SARS-CoV-2 reactive T cells, because they are also generated even in the absence of SARS-CoV-2 specific antibodies. So for example, in this study here from Sekina et al. From last year, who analyzed T cell and antibody responses in various patient groups, you can see that T cell responses were actually detected more frequently compared to virus-specific antibodies, particularly in cases of mild infection here. So importantly, also the analysis of SARS-CoV-2 specific T cells can also be used to monitor long-term immunity, be it after natural infection or also after vaccination. And also in particular, in regards to vaccine development, it's also important to study the implications of cross-reactive T cells for all the upcoming clinical trials that investigate the efficacy of the novel COVID-19 vaccines. And last but not least, also SARS-CoV-2 specific T cells can be generated as a treatment option, for example, in adoptive cell therapy. So, but how can we analyze antigen and virus specific T cells using flow cytometry? So, well, generally the methods can actually be classified into two main categories. So first, there are the direct analysis methods, which analyze T cell specificity via peptide MHC multimers. And these methods are really specific to detect antigen and virus specific T cells and they work very good for CD8 positive T cells, but only limited for CD4 positive T cells. They also do not provide any information on the functional capacity of the T cell subset, and also work limited only with defined peptides and MHC molecules. Second, there are the so-called indirect analysis methods, which more or less focus on the analysis of various effector functions. So, for example, the expression of certain activation markers such as CD69, CD137, or CD154, or also cytokines like interleukin-2, TNF-alpha, or gamma interferon can be analyzed. Of course, also proliferation or cytotoxicity, for example, could be easily analyzed using flow cytometry. In contrast to the direct analysis methods, the indirect methods actually work very well for all T cell subsets. They provide T cell subset specific information and are mostly independent of the investigated pathogen or also the MHC status of the donor. However, most importantly, the biggest advantage is here, of course, that all the relevant information can be obtained using flow-based assays. And at Miltenyi, we actually offer a complete workflow for the analysis of antigen and virus-specific T cells. So let's just have a quick look at the workflow. Of course, first, the T cells need to be isolated from a donor sample, and this could be, for example, tissue, blood, or also PBMCs. For the efficient tissue dissociation, we have the GentleMux tissue dissociator, which works really well. Following the isolation, the T cells need to be stimulated with the antigen of interest, for example, our peptivator peptide pool. And for stimulation, it is really important to know that you need antigen-presenting cells to be present, because they actually present the antigen to the T cells and also further activate them. When you work with PBMCs, for example, the antigen-presenting cells are already present within the cell populations. However, with isolated T cells from tissues, you need to add the antigen-presenting cells separately. Following the stimulation, only the antigen-specific T cells will be activated, and the cells can then be easily stained intra and extracellularly, for example, to assess their phenotype or also analyze cytokine expression. Before proceeding with flow analysis, the cells could also be pre-enriched magnetically. And then finally, the cells are either analyzed directly via flow cytometry or could also be sorted further on a cell sorter. So let's now just take a look on tools that can be used for the staining of these virus-specific T cells after antigen-specific stimulation. So of course, flow cytometry is still the most widely used method for the analysis of rare T cells. However, even established methods like flow cytometry have certain limits and challenges, especially because virus-specific T cells can be extremely rare in certain patient groups. And these limits and challenges were already illustrated years ago in a very nice publication by Bacher and colleagues, where the authors state that despite its high sensitivity, flow cytometry is always limited by the number of events that can be recorded, as well as the staining background, which is typically between 0.01 and 0.1%. So these limitations really restrict the analysis of rare cells, like, for example, virus-specific T cells. Nevertheless, with modern flow cytometers, you should be able to detect at least some cells, but there might be certain conditions where, let's say, hardly any cell can be detected, and here our MUX microbeads could be used to pre-enrich T cells before flow analysis. Furthermore, to overcome high background staining and also enable a more high-resolution flow analysis of these rare T cells, you can use our recombinant reaffinity antibodies that were already introduced by Ormi a couple of minutes ago. And then coupled with VioDyes, our reaffinity antibodies really provide a very crisp and bright staining with a low spillover, which is especially good for multi-parameter analysis. So here we just listed several reaffinity antibodies that might be of interest when analyzing antigen and virus-specific T cells. And once you get the slides, you can just have a look through the table and then click on the links to find your favorite marker, or you can directly contact us, because we are always happy to help you with the design of your individual flow panel. So I've also brought you some exemplary data. And here you can see how we use our reaffinity antibodies to detect SARS-CoV-2 reactive T cells, either in a healthy donor or in a COVID-19 survivor. And in this example, we actually collected blood samples. The samples were then lysed with our RBC lysis buffer and then stimulated for 4 hours with our SARS-CoV-2 peptivator peptide pools, or left untreated as a negative control. To identify SARS-CoV-2 reactive T cells, so the T cells that actually reacted against the indicated peptivator peptide pool, we analyzed the expression of interferon gamma on all CD3 positive T cells. And as you can see, actually the staining of T cells with our reaffinity antibodies allowed a clear identification of SARS-CoV-2 reactive T cells in the sample of a COVID-19 survivor. So hereby we could clearly identify which patient was exposed to the SARS-CoV-2 virus. So, however, we not only have the reaffinity antibodies, but we also offer really convenient, ready-to-use flow analysis kits, such as, for example, the rapid cytokine inspectors, which enable a rapid multi-parameter analysis of activated human CD4 positive as well as CD8 positive T cells. We also have the MaxFlex cytokine kits available both for human and mouse samples, and they allow a multiplex analysis of several soluble cytokines in just one sample, and also work on every standard flow cytometer. And last but not least, we also have the so-called 8-color immunophenotyping kit, which enables a flow-based evaluation of various leukocyte subsets, such as, for example, monocytes, neutrophils, TMB cells, as well as NK cell populations. And as already introduced by Omi, all these kits are also compatible with our very fast and convenient express modes that are available on our MaxQuant analyzers. And you can really imagine these express modes as some kind of software tool that will help you to automate your flow analysis. So with that, I would like to hand over to Jessica now, who will present our all-in-one solution for the analysis of SARS-CoV-2 reactive T cells.
Thanks a lot, Lorenz, for handing over to me, and welcome to everyone. I will now give you insight in our all-in-one solution to measure SARS-CoV-2 reactive T cells. Just give you a short overview about 3 different types of assays that were frequently used to measure immune response to SARS-CoV-2. The measurement of interferon gamma is often used to assess virus-specific T cell response. For the quantitative measurement of interferon gamma in supernatants, serum, and plasma, the ELISA technique is often used. ELISA determines the total concentration of secreted proteins after stimulation, but gives no information on single cell levels. Using the ELISpot, you already gain a few more information. This method is used to determine the number of cells secreting a cytokine after stimulation with a specific antigen, for example. Every interferon gamma secreting cell then gives an individual spot, and the number of spots increases proportionately with the strength of the immune response. The third method that you can see here is a flow analysis. Immunophenotyping using flow cytometry has become the method of choice for many researchers to identify immune cells in blood samples. By using multiple antibodies, you can differentiate between T cell subsets, for example, access activation marker, or measure cytokine production on a single cell label, as Umi already mentioned. These analysis techniques give the researcher the ability to track complex immunological changes. And based on that, we decided to use flow cytometry as a method of choice to design our all-in-one solutions for the precise analysis of SARS-CoV-2 reactive T cells. So here, let's have a closer look to our SARS-CoV-2 T cell analysis kits. Our portfolio includes 2 different types of kits to give the researcher full flexibility in the choice of starting material. The SARS-CoV-2 T cell analysis kit whole blood allows you directly see the measurement of T cell response in whole blood samples, while the SARS-CoV-2 T cell analysis kit PBMC gives you the option to measure T cell response in fresh, but as well as frozen PBMC samples. But however, both kits were developed for the same purpose. They were specifically designed for the precise analysis as well as rapid detection of both CD4 as well as CD8 positive SARS-CoV-2 reactive T cells in human samples. With all buffers, also the reagent and antibodies already included, the kit is the perfect solution for the phenotypic characterization of SARS-CoV-2 reactive T cells by flow cytometry. The kit is well suitable to investigate and also monitor SARS-CoV-2 specific T cell response in samples of individuals after infection or also after vaccination. This gives you the opportunity to assess the efficiency of different vaccines directly or even months after vaccination. So both kits have almost the same workflow, proceeding with the preparation of starting material for stimulation, following staining and subsequent flow cytometric analysis. I will now go into detail and will explain the workflow and highlight the difference between the two kits. So, as already mentioned at the beginning, we have developed two different kinds of kits to give the researchers the full flexibility in their choice of starting material. While in the SARS-CoV-2 T-cell analysis kit whole blood, we can directly start using whole blood samples. Our PBMC kit is designed for laboratories that work primarily with peripheral blood mononuclear cells, short PBMCs. So PBMCs are the most commonly used source because they contain both. They contain the antigen-specific T cells and the antigen-representing cells, and providing a functional immunological environmental context for the analysis of antigen-activated T cells. So normally PBMC are prepared by density gradient centrifugation. And when working with, for example, contaminated samples or infected samples, or also with small numbers of target cells, density gradient centrifugation can be very inefficient or maybe even dangerous. And these are all very important points to consider during our COVID research. So for the safe and easy isolation of PBMC from blood samples without gradient centrifugation, the MaxPrep PBMC isolation kit human was developed. This kit can be perfectly combined with our SARS-CoV-2 T-cell analysis kit PBMC. It performs reliable for every separation and keeps user inconsistency lows. This helps to further standardize your T-cell assays. So please follow the link here to get further information to the kit. So after preparation of the starting material, stimulation of SARS-CoV-2 T cells occurs. So the most efficient stimulation of T cells can be induced by using our Peptivator peptide pools. The Peptivator peptide pools are designed to stimulate both CD4 as well as CD8 positive T cells. Our standard Peptivator peptide pools are a mixture of a length of 50 amino acids that have an overlap of 11 amino acids to cover the complete sequence of the antigen. Our SARS-CoV-2 specific products are based on proteins of the coronavirus. So SARS-CoV-2 specific peptide pools are available for the membrane glycoprotein, the nucleocapsid protein, but as well as the surface protein. Peptide pools M and N cover the whole sequence of the respective protein, whereas different variations of the peptide pool for the spike proteins are available. We have Peptivator pools which cover different parts of the spike protein, as you can see here on the right side. Newly available is the Peptivator SARS-CoV-2 S complete, which covers the complete sequence of the S protein or of the spike protein. And following ordinary viral evolution, SARS-CoV-2 also accumulates mutations over time, and this leads to new virus variants. Therefore, we also offer Peptivator peptide pools covering the mutation sequence of the new SARS-CoV-2 variants. So please just follow the link to get more and also detailed information to our peptide portfolio. It should be also mentioned here that we have released several SARS-CoV-2 analysis kit PBMC that already include the Peptivator peptide pool. But however, for both kit variants, whole blood as well as PBMC, we also have kits available that require the Peptivator to purchase separately. This gives you, as the researcher, the flexibility to choose the right captivator peptide pool depending on your scientific question. Let's come back to our reagents, which are also included in the kit. To ensure that the cells used in the assay are functional and also responsive, our kit contains a reliable positive control cytostem. It has been developed for the rapid and efficient stimulation of human T cells by inducing the secretion of cytokines or the upregulation of activation markers within only a few hours. Additionally, during stimulations, all samples are supplemented with referee A to inhibit the transport of proteins to the cellular membrane and enhance intracellular cytokine staining. Limited to the whole blood samples is a subsequent treatment of the cells with red blood cell lysis solution, and this ensures the optimal lysis of erythrocyte. So, the next step in our workflow is the fixation and permeabilization of the cells, followed by staining. Our SARS-CoV-2 T-cell analysis kit includes an optimized staining panel consistent of highly specific recombinant or reaffinity recombinant antibodies for the pre-cell flow analysis. Here you can see a table with all antibodies which are already included in our kits. With the antibodies included in the kit, non-T cells are excluded and T cell subsets are identified. Additionally, for the analysis of SARS-CoV-2 reactive CD4 and CD8 positive T cells, the expression of cytokines and activation marker are assessed. Please note the staining panel in both kits has some minor differences. So the PBMC kit has a freeze channel instead of IL-2 to allow the addition of a specificity of choice. And in addition, the PBMC kit variants also include a viability marker. So after staining, the T cells can be then analyzed using standard flow cytometer, like for example, the MaxQuint analyzer. We have already generated some explanatory data with our kit that you can see here. So in this nice example, we stained PBMCs of SARS-CoV-2 reactive donors after stimulation with the peptidates that are indicated here. So for staining, we use the panel included in our PBMC kit plus CD69 to cover the free channel. For analysis, we pre-gated either in CD4 and CD8 cells. Then we assessed CD154 and TNF-alpha expression on CD4 positive C cells and CD69 and interferon gamma on CD8 positive C cells. As you can see, T cells of the recovered donor respond specifically to the stimulation with the peptidator. This example impressively demonstrates that the SARS-CoV-2 specific T cell response can be studied precisely with our kits.
Thank you very much, Jessica. Let's have a look now at the B-cell role in COVID-19 research. As mentioned before by my colleagues, we said it's more important to monitor T-cell response. In the case of B-cell response, in the past, it was a little bit underestimated what could be the role. That's because usually researchers monitoring disease or also immunization like vaccination tend to look at the antibodies in the serum, and I think this is enough to understand the immune response to a viral infection. However, last year, after a few months into the pandemic, it was very disappointing to see how individuals infected tended to lose the titer of antibodies in serum after a few months. And that's why initially there was a lot of disappointment, thinking that an immune response against SARS-CoV-2 is actually not possible to amount properly. However, some more recent publication beginning of this year have shown that both B cell and T cells can actually persist in the blood of the convalescent individual that have recovered from the disease. And so having that in mind, we thought it's really, really important to really monitor the immune response, also looking at the B cells. And to do that, what we have used is the recombinant protein of the SARS-CoV-2 virus that we have produced over the year 2020 in-house using a eukaryotic recombinant system. What we have produced was basically all the protein of the SARS-CoV-2 virus, including also the target protein on the virus on the host cells, the ACE2 protein, and also the spike protein in every form it could be useful to study it for. So this in the trimeric form, in the monomeric form, with the S2 and S1 domain, and also the RBD domain. In particular, in the last months, we have also started developing the mutation, the most relevant mutation that came up during the pandemic for the spike protein monomer and also for the RBD. And also a very important point for the next slide that you need to keep in mind is that every of this protein is available alone or also as with a biotin covalently linked. So how to use protein to study B cells? Well, it's really important to use protein because the B cell receptor recognize actually the full native protein properly folded and glycosylated. And to study it, what we have developed was a tetramer solution. Basically, it is quite easy to put together. You need to take a streptavidin that is already conjugated to a fluorochrome. There can be PE, APC, or different fluorochrome available in our catalog. And then 4 times a protein, for example, a spike protein or RBD domain that is already conjugated to the biotin. Biotin binds to streptavidin in a ratio of 4:1. So once you put them in solution together, 50 minutes room temperature, you can create the so-called tetramers. And once these tetramers are added to a suspension of B cells, then you can easily use this system to measure the presence of B cells via flow cytometry. You can check out our protocol online and also in the datasheet of our kits. So in all our experiment, we started from a pre-enriched population of B cells that we have collected from PBMC of convalescent SARS-CoV-2, so COVID-19 individuals, 3 to 6 months from the infection and the development of the disease. From the PBMC, as I said, we enrich B cells with our really CD90 microbit kit, which allow to have label-free B cells. And after this enrichment, we have done then 2 separate type of experiments. On one hand, we have done flow cytometry analysis, also adding the tetramers and other type of antibodies for the phenotyping of the B cells. And not only we have done flow cytometry, but also flow sorting, as I will show you later, and also culture of the cells. And in parallel, we have also used the same system with an anti-P microbit to enrich the B cells via magnetic separation. So let's look now at the results for the detection and phenotyping of the spike specific B cells. Here, for time reason, I show you only the spike results, but we have done the same experiments with similar results for the RBD. So one important aspect to keep in mind is that the tetramer we have used, we prepared them in 2 different colors. So one in PE and one in PE Bio 770, as you can see here. The reason why we prepare it in this way is because we wanted to have a double staining for the same protein over 2 separate channels. In this way, after gating over different important aspects like excluding doublets, only on live cells, on the B cells, then we have gated plotting both tetramers colors in the dot plot. And then the cells have been identified by looking at the diagonal, so by gating over the diagonal. This is an important point because it has been also used a lot in most recent publication of SARS-CoV-2 B cells, but also in the past, because really helps to only select the specific B cells for the protein and not background, let's say, cells. And as you see also from the panel, we have also added CD27 and also anti-IgE, IgA, and IgM antibody so that we could further analyze the memory B cells and the isotype specificity. As you can see also from the right side of the slide, we have tested this system on the convalescent individual versus the healthy controls. And as expected, it was statistically significant to see that the healthy control show a very low amount of antigen-specific B cells for the spike and for the RBD. And on the opposite, the convalescent individual had presence of these cells as expected, which confirmed that our staining is specific. The next step was to flow sort the cells, and in this case we have used the max quantitative cell sorter for a series of reasons I will show you in the next slide. As you can see, we have used the same panel actually for the gating, and then we have analyzed the cells after separation. As you can see here, we have reached a very high purity of spike-specific B cells. And on the contrary, for the healthy control, we didn't observe, of course, many of these cells, so it was not possible to sort them. Furthermore, we have also put the cells in culture, as I will show you in a second. So why using the max quantitative cell sorter? Important aspect to consider when choosing a sorter for your experiments. And one of them could be the safety. In the case of COVID-19 samples, of course, this could be a very important point because an aerosol could cause for operator or carry over also, it could be a problem for the further sample you're processing in the same sorter. And the mass quantitative cell sorter offers a solution for it because is based on the closed cartridge system, which is single-use disposable and is also aerosol free for the way it works. And in this way, you really have a so you reduce at the minimum, or basically you have absolutely no carryover, and you reduce really at the minimum the risk for the operator. And that's why it was actually used in several studies on the SARS-CoV-2. And another advantage of the max quantitative is that the way this technology works is really gentle to cells. And in the case of such a rare and fragile population, such as antigen-specific B cells, is the perfect solution. As I mentioned, we also put the cells in culture. So we have used our B-cell expansion kit, which consists of CD40 ligand multimer kit and also alpha cytokine, which allow a polyclonal expansion of B-cells. So we have taken the sorted population, so the spike-specific B-cells here, and also the spike, so all the other B-cells from the negative fraction, let's say, of the sorting, which we call here spike negative. And of course, both of them expanded in culture because, as I mentioned, this was a polyclonal stimulation. However, when measuring the supernatant of this culture for immunoglobulins, so antibodies specific for spike, we have observed that only the spike-specific B cells were able to actually secrete antibodies, which was exactly our goal. So to create a workflow able to then have antigen-specific cells able to potentially be producing antibody, for example, for therapeutic studies, for therapeutic antibodies development. And this is also very nice because proves that all our workflow is very gentle to the cells and is very specific and really allows to have at the end functional B cells. And in this direction, I just want to give you an overview on all the offer we have for the study of therapeutic antibodies, starting from preparation of the material. That is also very important. It can be either from the whole blood or from PBMC, as you can see here, where we offer a very long list of product possibilities for B-cell isolation. But it can also be from tissue, for example, using the GentleMax dissociator in case, for example, of study of B-cell specific for tumors. And then, of course, as mentioned, we have a lot of advantageous solution for the flow cytometry monitoring, like with the MAX1 analyzer and also the reaffinity antibodies. And as I also showed before, very nice solution for culture, including also other product like chiral ligands, for example. And last but not the least, the MAX1 Taito, as I proved before, is really the perfect solution for this type of research. And there's several advantages. Finally, let's not forget that after detecting candidate antibodies, it's also important to test them. And for that, we also have a very long list of solutions, for example, for NK, ADCC assays, or MLR, or antigen-specific presentation assays, and so on. Now I'm basically at the end of my part and almost at the end also of the webinar. I just would like to remind that actually all the protocol I showed you before for the flow analysis of antigen-specific, so SARS-CoV-2 specific B cells, is also available as a kit from April, so called SARS-CoV-2 B cell analysis kit for both specificity spike and RBD. And on the same direction, we also put together a kit for easy isolation or enrichment of SARS-CoV-2 specific B cells, also for spike and RBD. And with this, I would like to hand over to Urmi for the final slide.
Thanks, Christina. Thanks all my colleagues, and hopefully we were able to summarize how flow cytometry can offer the advantage for cellular analysis in SARS-CoV-2 research, with the highlight of our antibodies and our flow analyzer, which offers this highest reproducibility standard. You also heard from Lawrence the complete workflow, how virus-specific T cell play a crucial role in COVID-19, and how our solutions can be offered for sensitive flow analysis for this type of cell. Jessica also highlighted our all-in-one solution, the very nice SARS-CoV-2 T cell analysis kit. Hopefully that helps you to improve your standardization for cellular analysis, both from human PBMC and whole blood sample. Lastly, Christina nicely elaborated the analysis of SARS-CoV specific B cells, not only the detection and phenotyping, but also the sorting and cultivation of these cells using the SARS-CoV-2 proteins and antibody panels using our MaxQuant Titocell sorter. So hopefully this was a helpful webinar for you. Just as a further note for you, if you want to learn more in detail what solutions we offer, we have a very nice website. If you get the slides, you will be able to get directly to the link where we have combined all our solutions for not only cell analysis, but even beyond for the research of SARS-CoV-2. And also we have specific webinar highlighting the T cell solutions as well as B cell solution individually in case you are interested to learn more detail. And with this, I would like to just show the disclaimer slide. And finally, thank you everyone for joining us here today in the webinar.